All right, so we are now being recorded. So this is our 9th video teleconference in BIOL 104YC. In terms of schedule, we are doing this on the exact day, Wednesday. So we're back to Wednesdays. In this video teleconference, I would be reviewing a bit of DNA replication. And of course, we will move with our discussion. I will be discussing gene to protein, particularly transcription. And in the next video teleconference, that's March 24 next week, no, still on a Wednesday, I will be discussing translation. So we are going to cut that, no, so that it won't be too long for 1 hour. And then our exam, as I mentioned in the previous video teleconference, will be on the 31st of March. Okay, so we have ample time to finish our topics before the exam. So without further ado, let's us start with a review first on DNA structure and replication. So because this is a review, I encourage you to really speak up so that you can also review yourselves. Now, it's better to say what we have learned because in doing so, we actually remember more. So let's begin. We have here a series of pictures. You might want to view this from the left. Now we have top left going to the top right, and then bottom left going to bottom right, and then bottommost left going to bottommost right. So can anyone share to the group what's happening in the first two pictures here on top? Let me have my pointer. Let's begin here first. First two pictures. Anyone? Let's have this number 1 and number 2 first. Anything at all that you can say about these drawings or photos?
DNA, sir?
Okay, yeah, this is about DNA. But we started the discussion, if you remember in the video, we started the discussion of first discovering where really is the genetic material. Now, what are the two candidates? Can you remember what two molecules?
Nucleic acid and protein, sir.
All right, so in the chromosomes, as shown here in the first photo, these are chromosomes during meiosis. The chromosomes are distributed to the daughter cells. And so there was the idea that the genetic material must be found in the chromosomes. And so as you answered, chromosomes are made of two materials, protein and nucleic acids, particularly DNA. So the quest for finding out which among those is really the genetic material was born. So we start with this experiment by Griffiths. Okay, by the way, we are jumping experiments here. Okay, if you read in detail, there are actually more significant events, no, between these two events. But I just chose Griffith's experiment. So, what bacteria, if you can remember, anong klaseng bacteria? What's the scientific name of the bacteria that Griffiths used? Is it Streptococcus or Staphylococcus?
He used Streptococcus bacteria, sir.
All right, so this is Streptococcus pneumoniae, okay, which can cause pneumonia, no, to in this example, the mouse. So What is the conclusion of his experiment? Let's go there right away. What's the conclusion as shown here in the last picture? So there are two strains of Streptococcus pneumoniae, one that can cause a disease, the virulent, and one that cannot cause the disease, the avirulent version. So what is the conclusion of Griffith's experiment?
Kuan, sir. A mixture of the non-virulent strain and the heat-killed virulent strain can still kill the mouse, sir.
All right. So what he injected here, this yellow one that you see here, is the virulent strain, but it's dead. No? So theoretically, as shown in picture number 3 here, it should not cause a disease in the mouse and the mouse should survive. So nag-inject siya ng patay na virulent strain, but together with that, he also injected an avirulent strain. And then the mouse died. And then when he got blood samples from the mouse, he found out that there are actually virulent strains in the mouse. So where is that from? What's the term here? The genetic material from the virulent strain, even if it's dead, was transferred to the avirulent strain. What's the term? Starts with letter T in bacteria. When the character
Transformation, sir.
Yes, this is bacterial transformation, and this is very common in bacteria. Okay, they can easily pass their characteristics to another group of bacteria. No, so that's also mainly the concern, no, in antibiotic resistance. Like certain bacteria that are antibiotic resistant can actually proliferate, no, if you use antibiotics wrongly, no, not the right way of using antibiotics, you can increase the population of antibiotic resistant bacteria and they can actually transform other bacteria to become antibiotic resistant also. So that's because of bacterial transformation. But the scientific community did not really believe still that it's DNA that actually caused the transformation. So that later on came with Hershey's and Chase's experiment. Okay, so can anyone tell us a summary of this experiment, Hershey's and Chase's experiment?
About the protein coat, sir?
All right, so let's start with the protein coat. Can you elaborate?
Sir, again, sir?
Can you elaborate about the protein coat? Unsa may gihimo ni Hershey and Chase sa protein coat? What label did they use? Is it sulfur or nitrogen?
Nitrogen, sir? Ay,
Ah,
sulfur, sir, sulfur.
okay. So sulfur na ano no na a radioactive sulfur or an isotope of sulfur. So they labeled the protein coat of the bacteriophage with radioactive sulfur. And then what did they do next? Unsa man ni kani siya nga cell? Kani akong gisirkulan. What cell is this? Model organism. What bacterium? The one found in your gut. And also in the fecal matter.
They used kuan, sir, E. coli, sir.
Okay, so these are E. coli cells. So they infected, no, the E. coli with a sulfur-labeled bacteriophage. And then later on, they found out that the sulfur label did not enter the bacterial cell. So meaning to say the sulfur labeled the protein, so the protein was not the one that went into the bacterial cell. And so in the second experiment, this time they labeled the DNA inside the bacteriophage. They used, I think I had a mistake before. Ano ang ginamit nila? Not nitrogen, but I think it was phosphorus, no? It's radioactive phosphorus. Can anyone confirm if it's phosphorus or nitrogen?
Phosphorus, sir.
All right, so it's phosphorus na because DNA has phosphate groups. So they labeled the phosphate groups with a radioactive phosphorus, and then they let the bacteriophage na labeled with the phosphorus infect E. coli cells again. And then they found out later on that the radioactive labeled DNA actually went inside the E. coli. So this one closed the debate, no, onto which one is really the genetic material. Is it the DNA or the protein? So now we know it's the DNA, thanks to the experiment of Hershey and Chase. So now that that's closed, case closed, the next quest was the structure of the DNA molecule. So the first person who actually made a step into knowing the structure of the DNA molecule was a female. No, it was Rosalind Franklin. And what she did was she took a photo of DNA. Now, what did she use in taking this photo of DNA? Which electromagnetic radiation did she use?
She used X-ray diffraction, sir.
All right, so this is an X-ray, no, photo of DNA. So on the first look, it's not obvious, no. You cannot really. It looks like a letter X with something in the middle, but that doesn't really tell us anything, no. Layman, kita. Now we don't know how to read X-ray crystallography photos. We don't know how to interpret this, but of course the scientists know. No, they use an algorithm using a computer to interpret this. But unfortunately, Franklin died prematurely, so her work, she didn't finish this one, but instead it was found by her colleagues later on. And who were these people? So this is number 4. We are now on number 5. Who are these people who continued Franklin's work? We have the 3 Nobel Prize winners. Somewhere in the 1950s.
Watson and Crick, sir.
All right. Watson and Crick are the most popular ones, but they had a third colleague, si Morris Wilkins, no? So they built a model, a 3D model of the DNA based on Rosalind Franklin's X-ray crystallography photo. So that's why we now have our infamous model of the DNA, the double helix anti-parallel structure. And we also know that it contains nucleotides, and the nucleotides have the phosphates and the sugar backbone, particularly the oxyribose. And then the rungs of those will be made up of nitrogenous bases composed of T, A paired with T, C paired with G. No, adenine paired with thymine, and cytosine paired with guanine. Thymine and cytosine are both what? How do we call them? Thymine and cytosine are single ringed, and they are known as purines or pyrimidines.
They are pyrimidines, sir.
All right, they are pyrimidines, and then the double ringed adenine and guanine are the purines. So pyrines and pyrimidines always pair together, and they are bonded with a hydrogen bond. The number of hydrogen bonds between adenine and thymine are 2. No, dalawang hydrogen bonds as shown here at the bottom, while the hydrogen bond between cytosine and guanine are 3 hydrogen bonds. Okay, so that's a thing to remember. And there is also this discovery that yes, indeed, it's purine paired with pyrimidine. Otherwise, if it's pyrimidine, it will be too narrow. And if it's purine to purine, it will be too wide for the DNA molecule. Okay? And then, of course, we come to Chargaff's rule. Can anyone tell us what Chargaff's rule is?
Um, in summary, Sir Chargaff's rule is that the amount of pyrimidines present in a double-stranded DNA is equal to the amount of purines in the double-stranded DNA.
Okay, so that is a bit general, no? If we now come to thymine, the names of the pyrimidines and the purines, how can we state it?
The amount of adenine is equal to the amount of thymine, and the amount of cytosine is equal to the amount of guanine.
All right, so that's more specific, no? And that's actually found in your homework, no? In your problem set. So I hope that will help you analyze the problem set. So lastly, we ended with this. This is Michelson and Stahl's experiment on DNA replication. Okay, I don't want to detail anymore what they did. But what did they find out? How do we call the model of DNA replication? We have three choices. We have conservative, we have semi-conservative, and then we have dispersive. So which one is which?
It is more on semi-conservative, sir, because there is presence of an older strand and then the newer strand also, sir.
All right. So semi-conservative because the template will be an older strand and then the newly built DNA will be a new strand. No, so it's a hybrid of older and newer strands. Okay, so this is detail on the replication fork. Okay, so if you can remember the replication fork, the DNA will open, no, with the help of helicase, but to keep it stable also, para dili maglambod ang DNA in this portion, there is a topoisomerase. And to keep this open, there are also single strand binding proteins. So while that's open, a primase will start putting primers made of RNA. Okay, because the DNA polymerase is unable to start from scratch. So there needs to be an RNA primer. Okay, so once an RNA primer is ready, the DNA polymerase can then slowly add nucleotides in the 3' end. No, not the 5' in the 3' end of the DNA. But if you base it in direction on the older strand, it's a 3' to 5' direction, no, based on the older strand. But on the newer strand, the addition is on the 3' end. Okay, it's on the 3' end. So that's DNA polymerase. So we have two strands actually in this replication fork. Now we're dividing it into two halves. On the left half, the one on top is the leading strand because tuloy-tuloy ang paggawa ni DNA polymerase ng new DNA here. But on the bottom part here, this is the lagging strand because the DNA polymerase needs to stop and then go back. No? So starting with number 1, and then it will go back, it will go to number 2, and then it will go back, and then go to number 3, and then there will be gaps between these strands we call Okazaki fragments. Now, and those gaps are going to be replaced by another DNA polymerase, and the gaps between the DNA strands in the lagging strand will be ligated together by ligase. Okay, so question, which DNA polymerase elongates the from the primer? The main DNA polymerase, diba? We have numbers. Is it 3 or 1?
3, sir.
All right, it's DNA polymerase 3 now. So you can see here, DNA polymerase 3 is the one that extends the leading strand, no? And a majority of the lagging strand, DNA polymerase 3. Ang work ng DNA polymerase 1 is what? What is the work left for DNA polymerase 1?
Replication, sir. DNA replication.
Yes, both of them. All of this is DNA replication. Pero ang DNA polymerase 3 is the one that works straight, no? Many nucleotides in the leading strand and the lagging strand. So, unsa na lang man ang nabilin nga work for DNA polymerase 1?
Removing RNA, sir.
All right, it will remove the primers of RNA and replace them with DNA nucleotides. So, correct. Okay. And then the ligase will be the one to stick the ends together of the Okazaki fragments. Okay, so I hope you can remember that one. So this is only one half of the story. The other half is the other half of the replication bubble. Notice in the other half na ang taas na naman yung lagging and the one at the bottom is the leading. Okay, so there's a pattern. No? So please don't get confused by that one. And then if ever there are mistakes, it will be repaired. So usually we call the enzymes that would repair, they cut the portions that need to be repaired and replace them with correct nucleotides. We call them nuclease. So once cut, the replaced portion, the portion to be replaced will be replaced by DNA polymerase, DNA polymerase 1, and ligase will again glue together loose ends. This is DNA repair after replication. So usually, if you can remember, what is this error? The one that I'm encircling right now. Usually, which DNA nucleotide will get the errors? Is it the one with adenine, with thymine, with cytosine, or with guanine? Anyone? Wild guess. Kitsa man, si adenine, thymine, cytosine, or guanine? Who usually has an error or gets the error?
Cytosine, sir.
Dili. Another purine. Another pyrimidine. Kinsa man ang another pyrimidine aside from cytosine?
Thymine, sir.
Okay, thymine. So, usually kanina i-bond sa nga, this is a thymine dimer. Okay? And that's usually caused by radiation, or any other mistakes during DNA replication. So usually no, but not all the time. There are also other mistakes, but usually it's this thymine dimer. All right, and then we come into this telomeres, the ends of your chromosomes, wherein I highlighted last time there's a problem in DNA replication at the ends of DNAs, especially on the lagging strand, because as the RNA primer is removed, okay, take note the exposed end here is 5' and the DNA polymerase 1 cannot add to the 5' end. No, dapat 3' end. So this is left unreplicated, and in the next round of DNA replication, it's going to be cut short. Now, so the telomeres or the ends of the chromosomes get shorter as the replication progresses into several cycles. And scientists have associated that with aging. As you age, your telomeres become shorter. But there's an enzyme that prevents that from happening rapidly. The enzyme is called what? What's the name of the enzyme? It prevents the telomeres from becoming short prematurely. So telomeres, what's the name of the enzyme?
Telomerase, sir.
All right, telomerase. No? So thanks to telomerase, your aging process is not so fast. But still, telomerase cannot really just keep up all the time, no? So you will still age. But there are certain types of cells in your body that produce too much telomerase. And what are those cells?
Cancer cells.
All right, so cancer cells are known to produce excess telomerases, and therefore they are essentially immortal. If you just provide them with nutrients, they will continue dividing and dividing. Okay, so that's one strength of cancer cells. The other strength we discussed in cell mall is their excess of MMPs, matrix metalloproteinases, which allows them to invade other body parts, not to metastasize. So now let's talk about the next topic, transcription, gene transcription from gene to protein, but this time transcription first. Okay, so let's start again with an experiment, and this experiment was done by Biddle and Tatum together with their colleagues Serb and Horowitz. Horowitz. Okay, they used an organism, a bread mold. Now this is bread mold actually, called Neurospora crassa. That's the scientific name. So what they did is they grew Neurospora in a Petri plate, just like you see here, but then they exposed the cells to X-ray. So the X-ray caused mutations in the cell. Many of them did not survive the mutation. Many of them died. And Biddle, Tatum, Serb, and Horowitz only cultured those that survived. No? So they cultured it. So what did they do? They cultured it first in minimal media, meaning to say the medium here doesn't have any nutrients, no, and the cells did not survive here. And then they had another medium, minimal medium, pero they provided a nutrient valine, no. This is an amino acid valine. And then some cells have survived, some haven't. And then they also had a minimal medium, but this time only lysine. Notice here the cells didn't really survive. And then lastly, there's a minimal medium for plus arginine. And they noticed some of the mutants from their original mutants survived here. Okay. And their control, of course, is the wild type. What is wild type? This is the neurospora that they did not expose to X-ray. So the neurospora is able to survive even in minimal media, no, even with no nutrients, because they can create their own nutrients out of the raw materials. But among the mutants, as you see here, only the one grown in arginine really survived. Okay, so this is Biddle, Tatum, Serb, and Horowitz experiment. So what's the explanation behind this? Okay, so notice here, no, that in order to produce arginine, which is important in the Neurospora crassa, enzymes like enzyme A, B, and C should be present. Because what do those enzymes do? They turn a precursor, the raw material, enzyme A turns it into ornithine rather, and then enzyme B turns ornithine into citrulline, and then enzyme C turns citrulline into arginine. With the absence of any of these, your Neurospora crassa might not survive. Okay, and notice in this table, now there are three criteria of mutants. Mutant 1, class 1. Okay, ang class 1 na mutant will not survive in the minimal medium, but will survive in ornithine, citrulline, and arginine media. Class 2 mutants can only survive in citrulline and arginine media, while class 3 mutants can only survive in arginine media. What does this mean? So the explanation is here at the bottom. Now, in your wild type, all the enzymes are present, so no problem. But in class 1 mutants, they lack enzyme A. So meaning to say, if you grow them in minimal media, they cannot turn the precursor into ornithine, no? X. So they cannot produce arginine in the minimal media. So they will die. So they have to be provided first with ornithine, citrulline, or arginine in order to survive. That's class 1 mutants. Class 2 mutants lack enzyme number 2 that turns ornithine into citrulline. And so they can only survive when provided with citrulline and arginine. And of course, class 3 mutants will only survive when provided with arginine because they lack the enzyme that turns citrulline into arginine. So dapat diretso na sila bigyan ng arginine so that they will survive, no? So I hope you can get this table, no? You can understand it. And the explanation of Biddle and Tatum and Serb and Horowitz here is that the mutants lack enzymes because the genes that make those enzymes, remember enzymes are proteins, di ba? Okay? And proteins are made from genes, no? The instruction from genes. The genes that make those enzymes or that have instructions to make those enzymes are destroyed, no, in the X-ray because they were exposed to X-ray. So class 1 mutants do not have gene A. Class 2 mutants have destroyed gene B. And class 3 mutants have non-functional gene C. So therefore, the result of Biddle and Tatum's experiment. So before I move on to the next slides, are there questions on the figure here? Anything that you do not understand or you need more explanations of? I think this is found in your genetics book, but in a different presentation. Kasabot lang ba? Are these understood?
Okay, Russia, sir.
All right. Sige. So may kasamang hehe. So meaning to say, if I'm gonna let you explain, you will still pause many times. So I really hope now you the meat here, the conclusion is that genes, ito lang yung conclusion nila, genes help contain the instruction in making enzymes. That's the conclusion. And where are genes found? Anyone from the group? Saan nakikita ang genes? Where are genes found? Sorry, hindi ko nakuha yun. Wala gihapon, dili klaro. Can you type your answer? Where are genes found? Okay, I'm viewing the Google Meet app so that you can. Okay, go.
Ambasin na kuan yung connection, sir. Kuan in DNA, sir.
All right. So sa DNA nakikita ang genes, no? So from the illustration here in this slide, I'll go back to this. So, genes are found in DNA. Itong gene, this is from DNA. So when Biddle and Tatum and colleagues exposed their bread mold to X-ray, some of the genes got destroyed. No, mutation nahitabo. There's mutation. And because those genes are destroyed, the enzyme, which is a protein that they're supposed to be making, no, are also not present in those mutants. No, so you can analyze it that way. So a gene dictates for an enzyme. So during their time, their conclusion was that one gene is for one enzyme. But presently, today, we know that not all products of genes are enzymes. No, some products are hormones. No, hormones are not enzymes. Some products are structural proteins. Structural proteins are not enzymes. Some products are ribosomal RNAs, and those are not strictly protein enzymes. So during Biddle and Tatum's time, one gene, one enzyme. But now we know it's not just the enzyme that's a result of a gene, right? So let's continue. So the process that actually creates enzymes or proteins or whatever products from a gene involve these three. No? Transcription, that's from DNA to messenger RNA, that's transcription. And then there's translation from messenger RNA to your polypeptide. So this illustration here shows two scenarios, one for prokaryotes. Since prokaryotes don't have nuclei, their DNA is just floating in their cytoplasm, and the transcription can happen in the cytoplasm together with the translation. While our focus in this lecture is for eukaryotes, wherein the DNA is transcribed inside the nucleus, and it's the messenger RNA that gets out of the nucleus, and it's the one that's being translated using ribosomes to form polypeptides. So this is the model for eukaryotes. Okay, and take note, before transcription, there is before the end of transcription, there is RNA processing. So our topic this morning, now the rest of this lecture will be this portion. Inside the nucleus, DNA transcription and RNA processing. Right. So this, guys, I think you've done this already in your laboratory. This one is called the central dogma, no, of biology, wherein everything that you observed in living things, no, life forms, at least most of them, no, that are found in the cells, are created in this way from genetic material, genetic instruction, DNA, which is transcribed into RNA, messenger RNA, which is translated into proteins. And the phenotype that we are talking about, diba, the phenotype is the one that we can observe, is usually the protein, no, this portion, the protein. And the genotype, the one that we do not see, is the DNA. No? So for example, in Mendel, 'di ba, katong genotype na capital R, example lang to, small r. So let's say capital R is round and small r is wrinkled. Okay? Once this is already transcribed and translated, we can now see our phenotype. And the phenotype here is, what's the phenotype? Big R, small r. This one's going to be round. Diba? Kasi may isang dominant. So I'll just write it this way, round. Okay. So from genotype na DNA to phenotype protein, round. Okay, so this is a central dogma. And this is another illustration of the DNA with various genes color-coded. So you have gene 1, maybe the green one, gene 2, yellow, gene 3, the blue one. So these are translated. These are found in DNA, as you see here. Okay, and then they are transcribed into mRNA. Okay, take note, mRNA is single-stranded. No, so only one strand of the two complementary strands in DNA is transcribed. And which strand is that? Look at this. Alin ang basihan ng messenger RNA? Labeled na dito, it's a template strand. Don't worry, later I will have an example. No, we will have an example later. And then, of course, in the next lecture, no, in our next video teleconference, we'll talk about translation. So we'll cut it in transcription. So just a preview. If I'm going back here, The mRNA, no, the one that's being transcribed, the result of transcription, the mRNA, has what we call codons, no, in threes. And these codons, no, are read by threes, by three nucleotides in your RNA. So by threes talaga. And another thing to note is that the thymine in DNA is replaced instead by uracil in RNA. So there is no thymine in RNA. Please remember that one. So if we are now going to base on the codons on the mRNA, this is the codon dictionary. Now, this allows us to see what is the resulting amino acid with a particular type of codon. Okay. How many possible codons are there? There are three, diba? Three positions man. So in the first position, for example, yung UUU, no? How many possibilities in the first position? Anyone? How many letters to choose from in the first position of the codon? 'Ilang letra ang pwedeng pilian sa first position?
3, sir.
3 only? You have U, paired, you have U, you have A, you have C, you have G. So you have 4 choices no in the first position. So 4 choices. Okay. What about in the second position? Ilang choices? Can you repeat what you chose in the first position? Yes or no?
Yes, sir. Na parehara mo yung
Yes.
sir.
Oh, pwede ma-repeat. So ilang choices sa second position?
4 gyapon, sir.
4. So actually, all of these positions have 4 choices. So 4 times 4, 16. Times 4 pag yun, this is 64 codons that you see in this table. 64 yan sila lahat. But because one amino acid can be coded by more than one codon, for example, phenylalanine here can be coded by UUU or UUC, there are only 20 amino acids, no? But there are 64 codons. Okay, 64 codons. And out of the 64, one of them, AUG, is for start, no, beginning of translation. And 3 of them, UAA, UGA, and UAG, no, which we fondly call as UWA, UWAG, and UGA, no, these 3 are stop codons. So 64 minus 4, so 60 codons na lang left for the other amino acids, the 20 amino acids. But anyway, the detail of this we'll discuss next meeting. So let's talk now about translation. That's DNA to mRNA. So of course, it requires, as you see here, no, step number 1, there needs to be an RNA polymerase. So ang RNA polymerase will be the one to make messenger RNA. So as you can see here. So in step number 1, the RNA polymerase has stuck to a portion of the DNA. Okay? And what is important? What is in that portion? We have what we call there the Tata box, no, because it's made up of TA na sequence, no, sa DNA. So this Tata box is also known as the promoter region, no? And that's the indicator where the RNA polymerase will start. No, there's a Tata region. And not only that, transcription factors which are chemicals, no, important so that transcription will start, should be present also. So first, you need a promoter, that's the Tata box, only in eukaryotes. And then there's transcription, no, transcription factors rather. And once those are present, your RNA polymerase 2 can then start making messenger RNA. Right, so please remember that one. So the question now is this. you have many types of cells in your body. No, you have muscle cells, you have liver cells, nerve cells, blood cells, to name a few. But the instruction present in their DNA are actually all the same. No, the DNA in your muscle cell is the same as the DNA in your liver cell. How come the muscle cell is different from the liver cell? Can anyone answer that question? Nga pareha raman unta ang instruction in all of the cells of your body. Why is your muscle cell different from your other cells, from the liver cell?
They differ in terms of structure and function, sir, in the body, sir.
Structure and function. Okay, that could be an indicator. Okay, but that could be an answer, no, based on location. Any other answers? Genetical. Okay, continue.
Fibers sir na contain of hundreds of nuclei.
Okay, you're talking about DNA.
Yes, sir.
All right, so it has something to do with DNA, no? The first answer a while ago was something to do with location. Okay. Second one was trying to say, in terms of DNA, sir, you're both correct. Why? Why are they different? Okay, so let us answer that one. Now, take note of these transcription factors, no? Okay, so here's the answer. So notice here, all of the cells here have the same DNA, no? Yung blue line na yan. But look at the muscle cell. Look at the DNA in the muscle cell. The highlighted regions are different from the ones in the blood cell. From the nerve cell as well, different. Liver cell also different. Okay? Meaning to say, in the muscle cell, only the segments highlighted in red. No, let me have my pointer. In the muscle cell, only the segments highlighted in red are transcribed. Nakuha nyo? Okay, so back here. This is transcription, no? There are transcription factors. So the RNA polymerase 2 will transcribe, make a new messenger RNA. So in the muscle cell, only the two tips here in the middle are transcribed. While in the blood cell, only this upper portion here in pink is transcribed. While in the nerve cell, only this portion in yellow is transcribed. And in the liver cell, only these purple portions are transcribed. So same instructions, all in all, but they read different portions. So nagets ninyo? It's like providing you with the same book, but I'm assigning one of you to read chapter 1, another person to read chapter 2, another person to read chapter 3. No? So the different types of cells are reading different chapters of the DNA. That's why they are different. Nagets ninyo?
Yes, sir.
Okay.
Yes, sir.
And that begins with transcription, ha, itong transcription factors, no? In the muscle cells, iba yung transcription factor. In the blood cells, iba rin yung transcription factor, and so are in the other cells. Okay, so we're about to be done, so don't worry. So after that, no, initiation, there needs to be a promoter. What's in the promoter again?
The Tata boxer.
Okay, the sequence TATATA, no? Okay, in the DNA that needs to be present. There needs to be a transcription factor so that the RNA polymerase 2 will start. Step number 2, elongation. So the RNA polymerase 2 will move from 3' to 5' direction. So the oldest RNA is the one in the 5' end, and the newest is the 3' end. No? So the same as your DNA polymerase, it can only add in the 3' end. So that's elongation. So shown here in more detail, no? Only in the 3' end can RNA polymerase 2 add new RNAs. And then once done, no, there is a signal here na stop na ang transcription. The RNA polymerase 2 will go away, and you now have your RNA transcript. So your RNA transcript is actually not final yet, as you notice here, no? There are portions of the RNA transcript that need to be removed, okay? And we call those introns. So itong light orange, introns, intervening sequences. While the dark orange portions are the exons or the expressed sequences, no, of your gene. So those will be removed. Look at the final product here. It's a lot shorter than the original mRNA because the introns are removed. Only the exons are left behind. Not only that, but in the 3' end, a poly A tail, no, A is also added. And in the 5' end, a 5' cap, a guanosine triphosphate, as you see here, is added. So that your final mRNA is a lot shorter, no introns, and then there's a poly A tail, and then there's a 5' cap. Okay, and the enzyme that cuts the introns away is called the spliceosome. No, this one, the spliceosome. It's not really an enzyme, but it's a structure. The spliceosome contains sRNAs, no, or small RNAs that help in the locating of the ends of the introns so that they can be cut away from the exons. All right. So, question, why is it important to add the poly A tail and the 5' cap? Anyone? We're almost done, don't worry. Bakit kailangang may poly A tail and 5' cap? What will they do to help your mRNA? Walang answer? Okay. So remember, your mRNA is now going to go out of the nucleus. No? Here in the cytoplasm, there are a lot of enzymes, including nucleases. No? Because the cell is always prepared to destroy the DNA of foreign bodies. So when the mRNA gets out, it is going to be attacked by nucleases. No? And to prevent the instruction here in the middle from getting destroyed, the poly A tail and the 5' cap are added, no? Para even if the nuclease will start destroying the ends, it's just the poly A tail that's destroyed and not the essential instruction in the middle. No? So I hope you get that one. So that's the reason why there are poly A tails and 5' caps. Notice also here in bacteria and prokaryotes, there are no introns, no, walang introns, no. Here in the eukaryotes, there are introns that need to be cut by spliceosomes. Okay, so sample tayo, no, before I dismiss you. Nabay klase, guys? You have classes?
Non-Rasa ko.
Ang uban?
Wala sa ako, sir.
Okay, so sample, this is my last slide. So let us transcribe this, no? So this is your DNA. We are going to transcribe it. Notice that DNA has two strands. There's a template strand and a coding strand. So which among these do we transcribe? Is it the template or the coding strand?
The template strands.
All right, so the template strand. No, so let's copy the template strand. So we have 5', 3', we have A, G, T, T, A, C, G, C, G, A, C, A, T. Okay, there's our template strand. No, so this is a template strand. The coding strand is not transcribed. So now we are going to transcribe this. So the RNA polymerase Number 2, with the help of a promoter and transcription factors, no, sa naitata box, will start the transcription. So let me use another ink. Where will it start transcribing? From the 3' end or the 5' end?
3' end, sir.
All right, so transcription. So starting here, so I now have A, transcribe naman. Then paired with A, what's paired with A?
A, then U, sir.
Okay, so U, instead of B, it's uracil. And then C paired with G. And then
G.
this one is U, and then U, C, G, C, G, U, C, and then U. So in my mRNA, I have here starting from 5' and then 3'. Okay, that's now my mRNA. So this is now my mRNA. But there are still introns to be removed. What needs to be removed? So dapat ito mawala muna yan. No, that's an intron. No, look at this on top. And then G and C and U here should be removed. Okay, because they are introns. So my final mRNA is just U, C, A, U, G, C, G, U, G, U, A. And then this is my 5' end and my 3' end. So I now have my mRNA ready for translation. Okay? And if you look at here, translation proceeds from 5' to 3'. My first 3 letters from 5' are what? A, U, and G. And what does AUG mean? It means start. Okay, so there. So I have already transcribed my DNA here. So always remember, you transcribe the template strand starting from 3'. No? Another clue or another thing to note is that the coding strand is exactly the same as your mRNA, but the T's are replaced by U's. No, because wala namang timing ang RNA. Okay, so that's the example. Are there any questions about that one?
None, sir.
Okay, so very simple. No, in your next PS problem set, it will be about transcription and translation. So as of now, the reminder is still on your homework on DNA structure and replication. It's due this March 20, no, Saturday. So please finish that one. Right, so further questions, no, because we don't have any more time, you can just message me in eLearn or Messenger or email me, and then I will answer your question as soon as possible. So again, guys, thank you for joining in this video teleconference. The recording of this will be posted soon, and you can view it again. So see you in the next video teleconference where we will be discussing translation. So you are now all dismissed.
Thank you, sir.
Right.